Okay, good morning all. Hope you had a good weekend. I had promised a video posted for the Friday stuff that I wasn't able to deliver, and so I've made that video, and so I'm just doing a little processing, and so that'll go up onto our YouTube site, and a link from your Canvas will be provided here shortly after we complete the lecture this morning. Okay, so where we're at, let's get to it. So we are in 3, and so you're going to want to take a look again when I get it posted for you. I just finished doing a bunch of edits and whatnot, and so the video will be the first run through the Mapping Genomes chapter, and so my intro, you're going to want to see that. It's just about 30 minutes long. And so we're going to continue as if we have that already. And so, again, recommend, you read the entire chapter, right? Paying attention to your bold-faced words, et cetera, the section headers. But for emphasis, the material that I'm presenting is coming out of pages 63 through 72, and then 81 through 96 again. And in keeping with, again, using multiple sources, I'm hoping that you have access to your book from fall. And these are the chapters, 7 and 8, and then 18, 1 through 2, which has to do with sequencing. Those are things relevant to genome mapping. Okay, so again, what you'll find in the video that we posted or will post covering Friday work has to do with DNA elements and markers and explaining why maps are essential tools in genetics. And so where we're proceeding through chapter 3 is a little bit about linkage analysis, and remember to emphasize this is that this is the key to understanding how mapping works, is that linkage analysis stuff. And so you covered a little tiny bit about this in your intro genetics in fall. We'll do a little bit more this semester. We'll talk a little bit about the different kinds of mapping techniques, and then a couple examples of maps, right? All right, so off we go. So just sort of working kind of in inverse here, the kind of genetic maps that we're talking about are those that you generate through breeding techniques, which is a long way of saying that you have to have different and numerous kinds of offspring that differ in particular ways genetically, so that you'll be able to do a linkage map. Linkage map, if you recall, is the idea that you're relying on recombination between 2 points on the chromosome. Okay, and then how fine a scale the linkage map will be. So in other words, smaller and smaller and smaller DNA elements located to the genome in relation to each other. Well, that's going to depend on 2 things. Basically, you have to have a number of markers, and then you also have to have a number of offspring in which recombination has occurred, separating those, giving you recombinant types, right? Okay. So physical mapping then, so as opposed to genetic mapping, is that the markers are physically located to a point in the genome, and you make them visible in some way. And so there's techniques. Basically, the idea is a probe, right? You do a probe is a nucleic acid sequence of 70, 80, 90 base pairs long, and you label it in some way so that when it hybridizes, in situ hybridization, when it locates to its complementary sequence in the genome, because it's been labeled in some way, you can make it visual. Other kinds of maps then are cytological maps, and so we started off by talking about karyotypes, right, and using stains to reveal banding on the different chromosomes. Genetic maps restriction fragment length polymorphism maps. Those were first developed in the 80s and 90s, and these are based on the idea of using those endonucleases, correct? The restriction enzymes. A variant of the RFLPs are called AFLPs. AFLPs stand for amplified length polymorphisms, and so you do restriction cleavage and then you sequence and develop probes from that. So this RFLPs are not particularly dense maps, but this is a way of increasing the density by sequencing and generating probes. Okay, so then the other kinds of maps, and so instead of mapping, say genes, you can map other DNA elements, and so simple repeats of microsatellites, those are opportunities. Again, those DNA elements, you know what they look like, and so you can generate probes and you can map the genome for the simple repeats, the satellites. And then finally, and this is, I would guess, probably the end, if you will, of the mapping types, is the single nucleotide polymorphisms, right? So you have DNA elements in which you can compare individuals and you expect them to share the same sequences or differ by just simply one nucleotide. And so in terms of its density and the number of markers, obviously SNPs are going to be far more numerous and scattered throughout the genome compared to microsatellites or RFLPs. Okay, and then of course the ultimate genetic map, if you will, is to sequence an individual's genome. Genetic markers are defined as genes or DNA elements with known location in the genome. So that allows you to take a genetic sequence and to locate it to chromosomes, and so generating a physical map. Markers are used, of course, to map the genome, location of genes, okay, and locating them in relation to each other. But then genetic markers are also used to study the relationship between for inherited diseases, trying to locate a genetic difference. So you have a pedigree, and if you can get marker information for the pedigree and then compare it to other such pedigrees, so a pedigree of an individual who's affected versus a pedigree of individuals who are not affected, you can then develop a way of locating to the genome a difference between an individual who has a condition versus those who do not have the condition. Now, we're going to take advantage of that kind of genetic map in lab, and so that's, some of you have read ahead just a little bit on in the lab, and so you're seeing things like PheGenI database, in which we're looking up genes in relation to different phenotypes and whatnot. So that's kind of the impetus, the background of what we're doing, is we're using genetic marker approaches to study the inheritances of diseases. And then finally, genetic markers are, of course, famous in the law because, of course, we're talking about DNA fingerprints, right? And this now has a long history going back. The first genetic markers used in the law were, of course, blood types. Okay, so at any rate, genetic markers have a number of uses, and they're basically the idea is that we can compare individuals. We can compare individuals who have different disease conditions, as well as to understand where genes and other DNA elements are located in the genome to generate a physical map. So the basic premise of doing a map is the idea that the more markers you have that you can locate, so more markers, more loci, then of course the finer scale mapping you can get. And again, looking at the island of Oahu in terms of a map, right? That's the metaphor. So you can think of you know, how much detail do you need to put onto the map in order to know how to navigate from one point to another? So even though I've been here for many, many years, I still struggle with street names, for example. But if you talk about landmarks, boy, I know how to get myself around just about anywhere. And so, so for me, street names are not fine-grained enough. I need markers. I need, you know, where's the 7-Eleven? And that I can kind of map my way through the Honolulu and whatnot pretty quickly. But if you give me names, it's like, okay, all right, I got it. Because it's like I've lived in so many places over my life, and you may be thinking, oh, he can't speak Hawaiian words, and it's like, well, I do work on that all the time and can always improve, that's certain. But no, I'm just certainly pointing out that, like, for example, Bishop Street and, you know, and Young Street and First, Second, and Third Avenues. How many places do you think I've lived in which they had streets like that? So my memory is pretty good, but I combine maps from different places, and so it's landmarks that I use to sort of move my way around. So anyway, that's kind of the metaphor, right? So you're thinking about geographic mapping and then making the analogies to that with respect to genome mapping. Okay, so I need to know where the 7-Elevens are and where the McDonald's are or something, and I can kind of move my way around. So think about that as I have lots of markers that are scattered throughout Oahu, and so I can then locate myself quite well. If I was just to say use, you know, thinking of Chaminade University and University of Hawaii, Manoa, and West Oahu, and then maybe HPU. So I only have, if I was using colleges in their location as my markers, well, there's far fewer colleges than there are than, say, 7-Elevens and McDonald's. And so in using colleges as a marker, it's a very low density map, whereas using McDonald's and 7-Elevens, higher density map, easier to move around. Okay, so where we're at now, because of the human genome being sequenced, we now have the ability to compare individuals, not by sequencing their entire genomes, but just simply aligning and looking for single nucleotide polymorphisms. So you line things up between the genomes and you just simply notice the differences between them. So we have billions of points that you can compare, correct? And the thing is that individuals differ potentially at millions and millions of points along those genomes. And clearly then individuals who are related, so identical twins, would have very, very few of these differences. Siblings would have more differences compared to identical twins. Parents and their offspring, more differences still, and so on and so on. So you can start thinking about the amount of information given a pedigree. You can start doing some very fine scale mapping of markers to the human genome. But it all depends, if you recall from our discussion about linkage analysis, it all depends on having a pretty extensive pedigree. And so for many of us, pedigrees are not particularly extensive. Okay, so examples of markers follow. So phenotypic markers, right? So this goes all the way back to Mendel. So Mendel's factors turn out to be genes, and the different phenotypes, of course, correspond to different alleles. And so the first markers are discrete differences in phenotype. So there's lots and lots of these. This is, you know, a difference in digit size and dwarfism, and it turns out that this is often, not always, due to a single gene, a dominant mutation, and so you can relate the location of that gene by using an extensive pedigree analysis. Okay, The next level, again, we've already introduced you to cytogenetic maps. In many respects, you should think of these as phenotypes too, because the cytogenetic maps are based on using dyes to reveal the histone proteins that are associated tightly with the chromosomes. Moving past phenotype and then cytogenetic markers, we have the first truly genetic markers are proteins. And so we'll talk about this more when we get into proteomics, and hopefully those of you who are taking your biochemistry classes will spend a lot of time thinking about this. But so obviously there's thousands of different enzymes in us, okay? But coded for by different alleles, so the protein will have a difference of an amino acid or two, right? And so allozymes are the same enzyme but different alleles, okay? And then another way to think about this is that you have the same enzyme but it's coded for by different genes. And so allozymes, same enzyme, same gene but different alleles, and isozyme, same enzyme but different genes. We'll typically just talk about allozymes because distinguishing between the two requires a pedigree, and this isn't, we're not really talking and using these kinds of molecular markers that much anymore. But I wanted you to understand that just given a blood sample, okay, you can separate out proteins from the blood and you could run it on a gel. And once you deal with albumin, which makes a mess out of your gel, you'll get migration of proteins in the electric field, right? And then you apply stains, or you of course use antibody, a labeled antibody, in which case you can make the different proteins visible. Okay, so the very first one uses chemical means to make the enzymes visible, and this is a picture of a picture of a picture from the very first article, 1957, that used allozymes as genetic markers. And so they got families, 1, 2, 3, 4, and then they were comparing those individuals from different markers, right? And so anyway, it's really hard to see. And so what you're supposed to get from this is, okay, need better staining techniques? No, this was enough that they could basically show that individuals differed in predictable ways. So family members, they had the same banding, versus individuals who were not related, they would have different banding. Okay? And what's important about this, and at least in a historical sense, is that so geneticists, in particular those working with humans, you stop talking about sort of the normal versus the abnormal, and the idea that there's just sort of this one type of individual out there. So it's sort of the variation is substantial, and so the idea of the wild type, we use that except for just in the historical sense when we're introducing you to looking at fruit flies and pea plants, right? Okay. All right. So continuing on the way, so this is an example of a RFLP. So I forgot to mention, so another type of so the AFLP, that's there, the microsatellites and SNPs, those are all there, but so you'll end up, kind of visualizing them. So this first one here is a ladder, a size ladder, and so then these are different individuals, and then they show these different patterns, right? And so what you kind of got to go through is you compare. Oh, look, so there's an absence there. Compare. Oh, there's an absence there. And so you score individuals, presence, absence, for each one of these positions. One of my favorite markers is the idea of what's called the random amplified polymorphic DNA, or RAPID. And so what you do is you create random sequences as primers, 10 base pairs long. And it's like, well, what's the use in that? Well, we expect those just random sequences to be present in genomes. And then when you're comparing different genomes, you can expect that the random sequences, well, some individuals will have them and some individuals won't. And so this was if you don't have genome sequence, this is a way of interrogating and making comparison among individuals without actually having the genome sequence itself. Okay? All right. Okay. So how do you put this information together? So the idea is you have SNPs and you know the location in the genome in which these things are located, and then you can look to see, given a pedigree, so SNP1, SNP2, SNP3, and so on and so forth, you can start putting them together in terms of where they located in the genome. And so these form what are called blocks or haplogroups. And the idea is that they show what's called linkage disequilibrium, which is a fancy way of saying there's a correlation between the alleles at different loci in populations. Okay. So from this, you can generate each one of those columns represents a different person, and you can generate an SNP profile for your individuals. And so where this is headed is the idea is that if you can compare individuals who have a condition and individuals who don't have a condition, and if you see an SNP difference, that is giving you genetic evidence of at least a potential that they're different because of what's happening at that region in the genome. Okay, so some general characteristics of markers. So some things that you need in order for a marker to work really well. Obviously, there has to be variation in the population, okay? And the useful markers, though, shouldn't be so rare as to have been found only in one individual. So SNPs stand for polymorphism as opposed to the other version, if you will, of a single nucleotide variant. So polymorphism basically means that many individuals are going to have the rare type. Polymorphism means we can recognize a difference among individuals, and some individuals will have the same characteristics and others won't. Whereas a variant is a single difference that maybe is only found in one person. So they give you two slightly different pieces of information. Okay, so if you're with me, the ideal genetic marker is that it has variation, but the variation is sizable enough that you would expect lots of individuals to have it, lots of individuals not to have it. Okay. And then obviously you have to be able to distinguish between the two types. So if the markers are so close together, that would be one, you would need a fine, excuse me, that would be a very challenging genetic marker to use because you'd need a very large pedigree to be able to reveal the differences. And the other, of course, is that the allele, the difference between one individual and another, the product has to be visible. So what you want are codominant markers in which you can distinguish between the homozygous recessive, the homozygous dominant, and the heterozygote. Okay, so ideally you want 3 different types and not 2. So for example, dominant markers, there's only going to be 2 bands in the gel. You can't tell the homozygous dominant from the heterozygote. Okay, so I'm going to try to go through the linkage stuff again. All right, and so this just is again a review. We've got 2 genes, but more generally we have 2 DNA elements, and we want to ask where can we find them in the genome. And again, there's only two possible answers to that question. I'm going to ask you, so given a handful of genes, where are they located in the genome? What's the possibilities? So thinking just of two genes, there's only two answers. Either they're on the same chromosome or they're on different chromosomes. Okay? It's really that basic. You know, the next level, of course, is to say, well, if they're on the same chromosome, how close together are they on the same chromosome, right? But that's not the major distinction. The distinction is if you've got two genes, what are the possible locations in the genome? They're either on the same chromosome or they're not. And if they're not, that means you have to have two chromosomes. Gene A is on chromosome 1, gene B is on chromosome 2 or 3 or what have you, all right? And then, so of course, then that's the idea of the linkage. And this goes back to the Morgan-Fly group, right? So the genes are on the same chromosome, then we say that they're linked. The parental types would be big A and big B and little a and little b. And so what would be the recombinant types? In other words, that have been crossing over in gamete formation. Well, the recombinant would be big A and little b, big B and little a. New combinations not seen in the parental types. And the point being linkage, so the frequency of the recombinant types will be fewer and fewer the closer together the two genes are on that same chromosome. With the ultimate distance, if the two genes are on opposite ends of the chromosome or they're on two different chromosomes, right? The recombination frequency, which is the number of recombinants divided by the total, will be 50%. And in 50%, of course, what you're talking about is independent assortment. Genes are not on the same chromosome, they're not linked. That's Mendels' rule of independent assortment. Okay. So, reviewing, what does recombination do? It produces new allele combinations. Morgan's group was the group that came up with the idea of recombination and using recombination as a way of generating linkage mapping. You detect it by comparing, the output of genotypes from meiosis. What alleles do the gametes have? Contrasted with the input genotypes. Well, what you mean by that, or of course, what are the alleles that the parents have and what combination, right? So you're comparing recombinant types and parental types. So genes are arrayed in linear fashion on the chromosomes. The frequency by which alleles showed up in the gametes can be used to map genes. Those 2 points is why Morgan got a Nobel Prize out of it. And just like Mendel, some of the principles that Mendel discovered / thought about and presented, Morgan's principles are still applied today. Genes are arrayed linear fashion on chromosomes. One of the things that I should just say, Morgan's group got wrong was the idea that the arrangement of genes on chromosomes was permanent in the sense that generation to generation that you could always know that they would be located there. And so you should right away recognize that biology is much, much more interesting. And so meanwhile, Morgan was working with fruit flies. Meanwhile, others were working with different organisms, including Barbara McClintock. And of course, what Barbara McClintock is famous for is the idea of jumping genes. And that she, working with corn, was able to show back in the 1930s and 40s that this linear arrangement of genes on chromosomes, well, that's true. It didn't necessarily remain stable generation after generation. So linkage groups could be broken up. And that is the basis of the idea of recombination. Okay. Again, reviewing a little bit about Mendel, segregation and independent assortment. So you should relate segregation and independent assortment to the ideas of recombination and gamete formation. Just review kinds of stuff. I would expect you to know, what stage of meiosis that these two things occur, segregation of the alleles, okay, and then the crossing over, what phase of meiosis does this occur? Okay, let's see if we can kind of work our way through this. So we're going to have two markers, and the markers vary they each have two alleles, and we'll be able to tell whether or not they are present or absent. So this is just going to be what's called a dominant marker. We won't be able to necessarily tell heterozygotes. Okay. Two genes, each with the two alleles. Cross. What do you get? You get a heterozygote, correct? Okay, what are the possible gametes? And reminding you that two of these we saw before, the parental types. And two of these are new combinations, the recombinants. Expected frequencies, if independent assortment, equal. Recombinant divided by total, so the maximum is going to be 0.5, because of course the total includes the parental and the recombinant. And if genes are linked, then they're going to be substantially less than that 50%. Yields what's called a two-point linkage map. Crossing over, relevant person is Sturtevant. He didn't get the Nobel Prize, but it was his idea that the recombination frequency can be used to generate map distances. And we still use it today. The units are centimorgans, in honor of Mr. Morgan. All right. F1's two or more genes perform a test cross. Do you remember what a test cross is? The female is heterozygous, and the male is double homozygous recessive. The tester is important because they can only produce one kind of gamete, e.g. there would all be little a, little b, right? So we know then that any recombination must have happened in the female line. Okay, so this is an example from the fly lab. You've got red versus purple eyes. Okay, red being dominant, wing length normal versus vestigial. Normal is dominant. As it turns out, both of these traits are sex linked, meaning that they're on the X chromosome. Okay, genotypes of, here's my tester. No, excuse me, that's the parental lines, and so this is getting the F1s, and so the test cross, sorry, I got a little ahead of myself here. Here's our heterozygous female with our tester male. So then looking at the thousands of flies in the F2 generation, they associated the types. So this is a red-eyed and normal wing type. This is vestigial wing and white-eyed type, and these were the most numerous. And these were other types, and so this was a red-eyed fly with a vestigial wing, and this is a white-eyed fly with a normal wing, and so these were substantially less. So in reviewing, one of the things that I'm going to ask you to do is just to quickly tell me, is this a linkage problem, yes or no? And you should say yes, because of course these numbers are not at all close to each other. Well, these two numbers are relatively close, and these two numbers are relatively close. Clearly these are much different from this, so this is evidence of linkage. Because remember, our null hypothesis would be independent assortment, the idea that the two genes are on different chromosomes, and so therefore the recombination frequency would be 50%. And oh, by the way, you wouldn't be able to tell which are the recombinant types. What all you would be able to see is that these are roughly 25%, 25%, 25%, and of course 25%. little hint. I've noticed over the years that students sometimes, you know, look, because of the way experiments work, interpreting 151 versus 154, that's the same number. Okay? Well, no, there's a difference of 3, but it's like, well, when you're making, you've only got these 2 possibles, so these clearly are close enough to say that they're of the same type. They're the recombinant type. Now these, obviously this is a difference of, 100 or so flies, and so while the 2 numbers are not exactly the same, they're close enough to say that these must be the parental type. There can only be 2 types, recombinant and parental, when you have a 2-point cross. Okay, so that's the point here. I say that. Notice that the parental types, or the ratio is about 1, and the recombinant types, the ratio is about 1. So we put that, oops, got a little bit of formatting issues, but we put that together, the types, and we're looking at the recombinant versus the wild type. So I'll have to fix this slide, but here's our parental types, so there's no recombination between them, so we don't count them as evidence, but we do count the recombinant types, and we add them up and get 305 divided by the total, and so the linkage map distance in centimorgans is 10.7. And we would complete this problem, right, by drawing a line, because if you recall, that's the way we model a chromosome, just as a straight line. And then we locate the 2 points on that chromosome relative to each other. Since this is a 2-point mapping, it's like you don't have to show them close together. They could be far apart because we don't have any reference to compare them. Okay. All right, so let's move on to how do we do this with humans. So again, what we need to do is we need to find differences among individuals, and then we would need a pedigree. So we're interested in this person. Remember, when you have the arrow there, that's the proband, or that's the individual that comes to the attention of the medical folks, and so you would then generate a pedigree to figure out what is going on here. And so gathering information about the parents, and so you might have historical records, and so you can then map who has it and who does not have the condition, and then you start generating your molecular evidence, right? So you start looking at, in this case they are looking at simple tandem repeats, so satellites, and comparing the individuals in the where's my cursor? There it is. So individual 1, etc., all the way through. Remember, individual 1, that's that person there. This is the, 1, 2, 3, 4th generation. Okay, and so, where's my individual 1? Oh, I got myself lost. So this must be, so individual 1, 4th generation 1. Okay. Anyway, I'm sorry, I lost my coffee. Here's, each one of these is a marker, correct? And then they're going to differ for presence absence of the number of repeats. And so we kind of go through here, and these are the number of alleles, the number of repeats that they have, and we can compare who's got the same and who doesn't. All right, so here's 155, 155, 155, 142, 142. You see how this works. Okay, so this individual is heterozygous, homozygous, heterozygous, and so on. And you can and you start looking for regions that differ, that are associated with having the condition, versus regions in which they don't have the condition. Okay? So what's the difference? 155, 155. Oh, heterozygous. 226 versus 228 and 222. So that's a difference between this person and this person. Okay, so you're with me on this? I got a little, tongue-tied about identifying my generations. Okay, so very quickly you recognize that this is a statistical problem, and so they have a way of coming up with what's called an LOD score, or load score, and it's based on a simple logit algorithm. Okay, so logarithm base 10 of odds, and so the odds is you take the presence, absence, the frequency of those who have the condition and whether or not they have the SNP, divided by those who don't have the condition, and again the number of individuals who don't have the SNP. And so you do an odds ratio, and so that gives you the likelihood, a score of how likely the condition is associated with a difference at that SNP. Okay, so for two genes, if they're not linked, then the RF is 50%, and the odds of seeing a recombinant type then is 1 to 1. So 1 divided by 1 plus 1, so that's half. So the odds are half. Excuse me. Okay, so from pedigree to genetic hypothesis. So after it got the pedigree, the procedure is to make a number of estimates of the recombination frequency. You do this for each child, beginning with the firstborn, secondborn, etc. And then you take that and you calculate a load value, given all of those probabilities, and they add up and gives you a pattern of inheritance. And it's basically like saying that the two markers that you're looking at, well, you can think about the association between the condition and not having the condition with the marker. And so a load score that's greater than 3 when you do the calculation is evidence that the disease or the condition is linked to that SNP. A score less than -2 is taken as evidence against linkage. Now, let's see. So I'm going to show you the equation, and so I'll pause there for you to sort of freak out on me just a little bit. Okay. You had a good moment there? All right, good. So obviously I'm not going to have you calculate this sucker, all right? Pretty complicated thing. I just gave you the idea that it's those who have and those that don't. It's a ratio, okay? And so then there are some additional features of how you work with that, because you're taking the logarithm of the odds ratio. And so, of course, it's called, you use software for that, okay? So the point being, I want you to be aware that there are statistical angles to how you do linkage mapping in humans. You have to have a pedigree, and you have to make guesses about what the likely recombination is, and then you test that model to see how well it fits the data. All right, thank you, software. Okay, so let's go ahead and call that the end of this. We'll have one more lecture presentation on genetic gene mapping, and then we'll be moving on to chapter 4. So thanks for listening.